Hello, everyone. Good evening. My name is Rawi Nadav. I am an MD PhD student at the Technion studying in Professor Naama Geva-Zatorsky's lab. I study at the Faculty of Medicine and I truly enjoy every minute of it. It is a great honor for me to be your host tonight. For more than a century, the Technion Israel Institute of Technology has been a pioneer in science and technology education, delivering world-changing impacts. The Technion has earned a global reputation for its pioneering work in nanotechnology, life sciences, stem cell technology, water management, sustainable energy, information technology, biotechnology, materials engineering, and aerospace. It is also one of only 5 similar institutes worldwide that include a medical school, encouraging rapid progress in biotechnology, drug development, and stem cell technologies. Our first speaker tonight is Professor Naama Geva-Zatorsky, who is my personal mentor, principal investigator at the Technion Faculty of Medicine, the Technion Integrated Cancer Center. She is applying systems biology approaches together with microbiology and immunology to study the interactions between gut microbes on host physiology. The Geva-Zatorsky lab, established 3 years ago, studies the interactions of gut microbes and host immune system in health and disease. Research projects in her lab focus on deciphering how gut microbes interact with host immune system, which molecules are involved, and how can we harness the therapeutic potential of the gut microbiota towards human health, especially gut inflammation and cancer. She also studies antibiotic-resistant bacteria and the emerging field of the gut-brain axis, which explores the interactions between gut bacteria and the brain. Since the COVID-19 pandemic took hold, a group of researchers in Naama's lab, led by her, have been developing a coronavirus home test kit. The test is based on saliva samples and would be inexpensive and very easy to use, capable of delivering results in a short period of time. We will explore this project with her tonight. Our second speaker tonight is well known to our audience in the UK. Professor Hossam Haick is the academic chair of chemical engineering at the Technion, head of the laboratory nanomaterial-based devices. Professor Haick made his mark at the age of 32 with the development of the nano-artificial nose. The nanos uses nanosensors guided by artificial intelligence to detect the scent of more than 23 specific disease biomarkers in a person's breath, including various cancers, kidney diseases, Parkinson's, and Alzheimer's disease. As of August 2020, he was furthering the award-winning breathalyzer technology to pick up the novel coronavirus that causes COVID-19, even in asymptomatic infected people. Tonight, he will explain to us how this works. Since one of my PhD projects in Geva-Zatorsky's lab is a joint project with Haick's team, it's my personal pleasure to introduce them both this evening, as I see them both as mentors for me. I'm going to ask you to please enjoy Naama and Hossam's presentations, and remember to type your questions at the bottom of the page in the chat box. Enjoy your evening.
Thank you very much for inviting me to speak here tonight. It's a real great honor for me. And thank you, Rawi, for this very kind introduction. And I would add that Rawi, together with Nadav, led the project that I will talk about today. Also, it is, as Rawi says, a great honor to speak together with Hossam as we have a joint project together and more to come. So I will share my slides and start. So as Rawi mentioned, our lab is usually studying the gut microbes, the good bacteria that live in and on our body and their interactions with the immune system. However, as the corona pandemic started, we were at the lockdown phase. Our lab was locked and we had a hard time staying at home, and we brainstormed together with these amazing team that I'm fortunate to have in lab, starting to think, how can we contribute to this pandemic? we wanted to find the best way we can contribute to this pandemic. So by some researching and talking with doctors at the Rambam Hospital, we realized that there is a major challenge in diagnosis. The current standard diagnostic channel takes several hours, sometimes days. Managing the suspected patients is a big challenge. Sometimes patients are put together in a room. Some are positive, some are negative. The isolation rooms are reaching exhaustion very fast. So contagious patients can infect others. There are delays in diagnostics and in interventions and in admission of the patients to high risk and COVID or non-COVID wards. So we were thinking, how can we contribute, and thought that if we can develop a convenient test for diagnosis that can be easily tested, no need for hospital equipment or for prior professional knowledge, can be self-home testing. This can help for safer epidemiology, for safer screening, and so on. And we will talk about it again and again in this presentation. So this is how we looked 2 weeks into this work. As soon as we realized the reaction that I will present to you in a moment, we just couldn't sleep. Literally, we came to lab, this team and a couple more members that are not in this picture. We had others at home. We worked like a multi-brain, multi-hand, multi-legs machine. It was a really amazing experience. Not much sleep, not much food, but we managed with science and the motivation to make this happen. So I think you can tell from this photo how tired we were. I would like to mention that it was in a great collaboration with the Rambam Hospital, who also realized the potential and really did everything they could to make this happen as fast as possible, and hosted us in their Corona lab to perform these experiments. So our development is actually a PCR reaction, like we hear that is performed at the hospital, but it is isothermal, meaning this PCR reaction, although it amplifies the nucleic acid of the virus, it can be performed in one temperature. I should mention that at the hospital, PCR is performed, but it requires a machine with cycles, a more complicated machine with cycles of temperatures that go up and go down for the exact amount of minutes. Prior to the PCR reaction, there is an RNA purification step that requires professional experience and an equipped lab. We were interested to develop a protocol that is simple, does not require complicated professional machines, and that the results would be easy to interpret and hopefully with low cost. I would mention already that at the end of this, at the end of the first step of this endeavor, we were reached out by Dr. Oren First, who licensed the technology together with the Technion. And the company is named Rapid Diagnostics, and our isothermal detection is this invention. So the idea is for one to do it in clinics or in workplaces, as it can be performed also high throughput in these 96 well plates, which I have one here to show. And it can also be done one by one. And we're working together with Professor Moran Berkovich and Baruch Wolfson from his lab for a test that can be performed at home based on this technique. So the idea is to perform the detection from human samples directly from the saliva. So it doesn't hurt, it's very simple. I will show at the end how it is performed, but it's basically this loop that goes into your mouth, not deep at all, just a grid on your cheek and then the saliva pool in the mouth, and then into a tube. And after 30 to 40 minutes, we can see the change in the color of the reaction. So the reaction starts pink, and if, God forbid, you're positive, it becomes yellow. We all want to think pink and remain pink. The idea is that the reaction is performed inside this tube, and while the enzyme that is inside this reaction mix is amplifying the nucleic acid, protons are released. These protons reduce the pH in the reaction, and that turns the pink color to yellow. So this is the main principle here. What we've done is to take several steps really off the shelf. It wasn't a very long research to get to this first applicable step, and perform this directly on samples on patients. So we performed this at Rambam, and these are examples. We first worked with the regular samples that are taken at the hospitals. So how, as you can see, at time equals 0, the reaction is pink. And if you are positive, it becomes yellow. And I'll show you at the end the actual tubes here in lab. So negative remains pink and positive becomes yellow. That was on the actual swabs that are taken at the hospital, but we also performed it on saliva samples, and these are saliva samples from positive patients. So at the bottom, it's the PCR reaction at the hospital, showing these are positive and this one is negative. At T equals 0, the reaction is pink, and at T equals 35, the positive ones become yellow. So there is a clear difference between positive and negative. We published this study. As you can see, Nadaf is Rawi Nadaf, and the first author here is Nadav Ben Asa, and I can also send you the links for the papers or for the news. The important part that I wanted to mention is that we had 0 false positives. So I'll talk about the false negatives as well, but as of today, as of now, we tested hundreds of samples, and we don't have any false positive results. So if you are positive in this test, most probably you are positive. We still recommend at this point to take the regular test once you become positive. So these are our results on hundreds of swabs. The y-axis here is positive in our test, so yellow, or negative in our test, pink. And the x-axis is the actual result at the hospital. So at the hospital in Israel, if you get a result of 35, you are considered positive and lower. However, several research articles have recently shown that contagious phase is under cycle 30. The limitation of our reaction is that we can detect if you're positive only at the contagious stage. So I don't know how much of limitation it is. It is a limitation according, if you compare it to the cycle 35 of the hospital. However, we detect you if you are contagious. So, and there are thoughts, at least I know in Israel, whether this number should be reduced as it admits people that are not contagious to the corona ward, which they encounter with contagious patients. So at this range of under cycle 30, we have sensitivity of at the current stage today of 94%. So 94% of our samples are true positives, and we have 100% specificity, which we tested both on other viruses and also by the fact that we have 0 false positives. So I hope this is clear. The company is routing for two directions. One is for high throughput testing, meaning these 96 well plates or 384 plates with robots at the lab that will enable cheaper, faster testings for workplaces or for whoever is interested. Also, a high throughput testing at workplaces, at the workplace itself, as it can be performed with very simple equipment that I will show you at the end. Rawi will help me. And the second direction is home testing, which is with this device that we are now developing in lab in collaboration with Professor Moran Berkovich and Baruch Rothman. We did go to India for together with the Ministry of Health and together with the Israeli Ministry of Defense to test 5,000 patients over there. So at that point, we were interested to collect the saliva and test it in Israel. So this is our freezer with 5,000 saliva tests, and we performed to date hundreds of them, which enabled me to say with confidence that our false positive rate is extremely low and actually zero, and also enabled me to say with confidence the 94% sensitivity. So the idea is that this technology is disruptive, decentralized, enables rapid and low-cost saliva-based detection with high specificity and high sensitivity, high throughput, and at home. We are also developing these days an app that would allow both to show that you're the one that is testing yourself, and also for easy detection of the results, if just to make it easier, although it's very easy also to see this pink and yellow. So it's rapid, accessible. The idea is to have this accessible. We most probably will start at the Technion very soon, and with some airlines and even with hospitals to eliminate bottlenecks. So at the Rambam, the interest in this technology is because at this point, it takes several hours until the patients receive the result, and they might be in the same room during this time. So at least the positives ones at our test can immediately go to the corona ward, or at least to an isolation room, and until they get the actual cycle from the hospital. It is dynamic, so it's a platform. At this point, it's on SARS-CoV-2, but it can easily be applied to other pathogens, either bacteria, either viruses, and also can be multiplexed, so detecting both influenza and the common flu and the SARS-CoV-2, the COVID-19. It's scalable, no need for prior professional experience, no need for RNA purification steps, and that is why there's no need for professional experience, and it's still a molecular test because it actually detects whether the virus, the nucleic acid, the DNA, this virus is an RNA of the virus is present. So these are, as you already know, Rawi, these are the two publications Nadav and Rawi have published lately in peer-reviewed journals, and would mention again that it is a collaboration between the Technion and Rambam. So thank you very much for listening, and I'll show you the test. Rawi will help me here. So it's in this tube, you have this loop. You just put it in your mouth, not too deep at all. And then you add it, to the tube. Oops, sorry. And that's it. And this tube, the loop can go back to this tube and trash. And this tube goes to the heater, which is preheated to 65 degrees. And that goes for 30 minutes. The result, so These are control, positive control and negative control, but positive control not for SARS-CoV-2, for human DNA. So this is yellow and pink, and this is maybe easier to see in these tubes, which are prototypes. So I hope it is clear, and I'd be very happy to answer questions.
And thank you, Naama, for the very fascinating invention from your lab. And thanks for your own contribution together with your team for the whole fight against these pandemics. Together with me in the lab, I have Ilyas Mansour, who is a PhD student in my lab, and who will help and assist in demonstrating the device afterwards. So I would like to thank Naama for the introduction. So I will skip the introduction about the need for fast diagnosis of the COVID-19, as well as for the challenges in front of the diagnostic tools that we currently use, such as the PCR and the RT-PCR. So in this regard, our approach has been different a little bit in the sense that we want to provide a very fast diagnostic tool that can enable the results within 30 seconds, and to do it in a non-invasive way. So one can use it at the gates or supermarkets or airports or at home or almost everywhere. And in the sense that this device could be used in a way that any person, any layman can use it without any experience. So we can save the logistics. So just to remind you, the two major challenges with the current available techniques are the logistics, which slow down the access to the results, and the other one is the low sensitivity. And there is another issue, is the fact that most of the patients with COVID-19 are asymptomatic, in the sense that they have the coronavirus in their human body, but they don't have any side effects, or they don't feel side effects. And as such, it's very difficult to suspect their health conditions, and therefore it's very difficult to do any examination for them. So our solution for this issue is to provide a portable device into which the person can exhale for 2 to 3 seconds only. The device then will interact some biomarkers in the exhaled breath that can result as a function or as a result from the coronavirus existence. Once there will be an interaction between these biomarkers in the exhaled breath and an array of sensors that we have developed and patented in my group, the results will be translated to digital signals that we analyze with the help of artificial intelligence in order to know what are the conditions of the health of that person, and what are the subcategories of that group of person, whether the person is healthy or has a disease. In case the person has a disease, what is the stage of the disease? And in case of being healthy, whether it's asymptomatic or really healthy without any suspectable conditions. The overall idea which I would like to speak about is based on the emission of volatile biomarkers from the disease place. In this case, I would like to demonstrate the issue of the cancer, of course, not the COVID-19, but it's the same principle. So once a disease starts to develop inside the human body, it starts to emit some volatile biomarkers, namely those small molecules with high vapor pressure that emit into the blood. And because they have low molecular weight, they are exchanging along the alveoli in the lungs, and they appear in the exhaled breath at very low concentrations that range between few ppbs, which is part per billion, to tens of ppb. When I say ppb, just to demonstrate the challenge, it's one part of the biomarker of the disease that is surrounded with 1 billion other species of products that doesn't or don't relate to the disease per se. In other words, it's like to try to look for somebody in China without any prior knowledge about his existence and its location. In order to do so, we have run earlier in our early studies on in 19 clinical centers worldwide, some examinations on real patients, and we have found that for every person there is a unique chemical print that appear in the exhaled breath that could distinguish the healthy person from the diseased breath by the composition of chemical compounds that exist in the exhaled breath. For example, if you take a look on the x-axis here in the graph, you can see that for each of these 17 types of disease, there is a unique combination of colors, which is unique combination of composition of compounds in the exhaled breath. And as you can see, for every disease, there is a unique fingerprint that is different than the other type of disease. And this is not only different from one disease to the other disease, it's also different between the subcategory of the disease, including the stages of the disease, whether the person has early stage of the disease or advanced stage of the disease. Nevertheless, all of these results we have achieved with the so-called mass spectrometry, which can be seen within a minute in our labs, which is a really very big device. It's like this device which you see on my back. This is a huge device. It costs around $500,000. It takes around a few hours to analyze one sample, and so on. And therefore, it can be used for research purposes, but not for POC purposes. And as such, we have taken this evidence, which we have proven for the first time since 2,400 years, and we tried to develop with it a device which can collect this signature of the disease and to detect those in portable way, as I mentioned earlier. And in this case, what we do is to capture all of these biomarkers of the COVID-19 with an array of nanotechnology-based sensors. Once these biomarkers will interact with the surface of the sensors, they will generate an electrical signal as a result of the chemical interaction. And all of these signals together, we feed into artificial intelligence, and we train the artificial intelligence, the signature of the COVID-19 versus the healthy controls and other types of confounding factors. With this approach earlier, we have shown that we can distinguish and detect 17 types of different disease, as Rawi has introduced in his own introduction. And as you can see here, we could distinguish between different types of cancer, such as colon, ovarian, bladder, head and neck cancer, pulmonary hypertension. We could also distinguish between neurological disease, such as Alzheimer and Parkinson, and multiple sclerosis, etc., etc. And as you can see, we could distinguish between all of these with really very high accuracies, mostly above 90% accuracy. And in many cases, such as in the orange color, we could distinguish them with accuracy that range between 80% to 90%. And all of that with the same device, with the same sensors. The only thing that we change between one application or one disease to the other disease is the algorithm, in the sense that one device can really do scanning for a wide variety of disease. With this device, on March 2020, and before we had the first coronavirus in Israel, we already sent our devices to Wuhan in China to do clinical study with COVID-19. And with this device, which we will demonstrate within a minute, we went into clinical study that included around 328 samples. And in that stage, we asked real patients and volunteers to exhale into the device remotely from 2 centimeter distance for 2 to 3 seconds. And then we have collected the results and analyzed the signals to see whether we could be successful or not. And here you can see part of the results which we have achieved. In order to, we were able to detect COVID-19 and to distinguish it from control with a sensitivity of 100%. In our case, what is most important is the sensitivity in our case, which is quite high accuracy. And if to be compared with the PCR, it's by 20% more and higher than the PCR, which currently in use in the clinics or the hospitals. Furthermore, very soon we will be in the winter season, at least here in Israel, and many people will have cold or influenza with similar symptoms to the COVID-19. And it's very important to distinguish between those people that have COVID-19 and those people who has lung infection that is not COVID-19. And as such, we have run also a clinical analysis with the real patients on people that have both health conditions. And as you can see, we could distinguish between these conditions with the sensitivity of 97% to 100%, which is absolutely an excellent result as well. So in other words, we are ready to the winter. Furthermore, it was very important to see whether people that had COVID-19 and recovered from COVID-19 would have signatures that will enable us to follow up those people, so we can either follow up with the treatment or with further diagnosis or with monitoring. And as you can see here, we have taken a group of people that had COVID-19 but cured from the disease. And as you can see, we could follow up the treatment aspects with accuracy that range between 92% to 100%, in the sense that the same device using different algorithm can enable us to monitor the recovery of the people using only exhaled breath with really very simple procedure. Something else that we are trying these days, even though we are collecting the data yet, but it's very important to update you. It's a project that has started with Bill Gates before 5 years on TB, on tuberculosis. And the idea was to develop electronic nodes such as that I have told you, but in this case to detect the disease in the form of a patch from skin. In other words, our aim was to develop a patch with really thin substrate that you can attach it to the skin, either in the area of the lung or other place. And that device can detect the disease from the skin rather than from breath. And of course, you can see one demonstration of these devices or patches, which is tattoo-like. And as you can see, this device includes 6 sensors, which we call it field effect transistors, that we have built in this form. The thickness of the device is 2 micrometer. This is the thickness of the device, and this is the diameter of the hair, in the sense that we have a thickness that is something like 20%, 20 times to 50 times less than the diameter of the hair, which you can stick it in your skin and it can diagnose the disease. The application of the patch on the skin is really very easy. You simply put it on a paper-like substrate and you just wet it with water, as you can see with this demonstration. Then you peel off the substrate, as we do with the fake tattoo, which we apply to the hands of our kids. And then you will have the device on the skin of the person. The device is quite stretchable and has a lot of other characteristics, and it can hold itself for several uses, even after scratches. So this device per se will be used very soon in clinical studies, but in earlier study that we have done with the Gates Foundation on more than 1,000 patients using analysis throughout skin with the offline mode, we have shown that we can detect another infection disease, which is called tuberculosis. And as you can see, the diagnosis accuracy which we could reach was between 92% to 97%. So our assumption was if we can detect tuberculosis from skin, we have higher probability to detect also COVID-19 from skin as well because of some similarities between the two types of disease. So with these two approaches, with the patch which we develop these days, and we will enter clinical studies, as well as with the breath analyzer, we do hope that we will be able to connect those to the cloud system. And with the cloud system, we can do also mapping every day. So we really can get epidemiology over the different sites in the world, as could be seen here. And also we can connect at-risk population with the doctor throughout IoT techniques and et cetera, et cetera. So I would like right now to move the video to Elias so he can show you the device and some demonstration in this regard. So as you can see, Elias will sits in front of a software, and right now he holds the device, and he will exhale as he will demonstrate. So now he's preparing the system and make some background measurement of the air in the room. He has exhaled for 2-3 seconds only into the device. And as you can see, we will have changes in the signals on the screen quite immediately. Hopefully you will see it. That's it. The device already processing the data, and within a couple of minutes, we will have the results. Of course, today we will not expose the result or the health condition of Elias. Can you please remove the cancel? This is the device which you can see. It has a remote sensor which can enable you, let you know how close you can come to the device. It can give you some indications, red and green, when to start the experiment. So everything is simple. And as you can see here, we have only one button inside the device. So it's quite easy to use. And one of the most important points that you can do it remotely. You don't need to exchange any disposable or any side accessory, in the sense that you can imagine to use it exactly as we use the thermometer when we go into into supermarket or gates, where they remotely sense our temperature from hand or from face remotely. And this is the same, we can use it. Hopefully very soon we will bring it to its destination into the market. So today there is a company called Nanos Medical that is taking this technology into the market. And of course, there are the challenges of the registration and the regulation of the health agencies, but this is something that is already in the hands of expert people. So hopefully we will see it soon in the market. Thank you very much.
Thank you both for these wonderful presentations. Very interesting work. We have some time now for questions. So if you the Q&A box in the bottom of the screen, you will be able to see the questions. I'm gonna go through them and pick a few to start with and see how much time we can have. If we start with the question for Naama about the HomeKit version of the device, will it have a heating device with it? And how is that going to be cheap?
Thank you very much. I'll answer, but first I'll say that I really enjoyed Hossam's talk. I'm always fascinated by Hossam's work and looking up to him. It's really amazing, really. So regarding the question, at home, at this point, before the development that I mentioned that we're working on, it can be done in a thermocup. We did it in a thermocup. So you need 65 degrees, but it's like a grandma recipe, as I call it. It's like plus minus. You don't have to be 65 degrees, 0.0. So what I showed here was temperature block inside. At home, I've done it in my oven. My oven can be set to 65 degrees. So baby's bottle warmer can do the work. So at this point, these are it's very versatile. We did not develop our own temperature block. We didn't see a reason to do that. But as soon as the device would be ready, it would be very simple, disposable, and everything all in one. So yeah, I hope I answered the question.
Thank you, Naama. I think now we're going to take a question for Hossam. So we have a couple of questions about how do you sanitize or disinfect the device between users. And there's another question about comparing between asymptomatic and symptomatic patients or infected people.
Okay, so I will answer all of these questions. The devices or the sensors are made from chemoresistors based on nanoparticles, which are coated with receptors, but in our case, chemical receptors rather than biological receptors. These sensors we can use up to 10,000 times before we replace them. In many instances, you can, of course, always go down to 1,000 or 2,000, but our capacity is 10,000. And we have to wait something like 1 minute to 30 seconds between one measurement to the other measurement in order to process the data. As you could see from the demonstration, the exhalation and the collection of the breath, it's 2 to 3 seconds only. But of course, we need some processing, which is up to 1 minute. So this is the time that is called dead time, that we have to wait between one device to the other device. Of course, this time period, we can reduce it down, but that will cost us in the accuracy. In terms of the cleanness between one measurement to the other measurement, as you could see, we just have no touch between the mouth to the surface of the device. But there will be on the surface some plastic that will be exchanged from one user to the other user. So we will not have any cross-contamination in this regard. And of course, the device itself has a one-way valve. So what is exposed inside the device will not come back to the breath of the other user.
So maybe we can take another question for Naama about I'm just looking for the question. The device isn't responding. Naama, maybe you can pick.
I can, yeah.
Question. It's just my device isn't responding. Okay.
There was a question about asymptomatic, and yeah. I think Hossam also answered. So on that question, it's it doesn't matter. Meaning, if there are asymptomatic patients that have high viral loads, so it works because it detects the virus presence. So since it's quite scary, actually, the paper showing that asymptomatic patients can have really high viral loads and are very contagious. Our detection method cares about the presence of the virus, so it can work also on asymptomatic patients. Yeah, maybe I'll turn it to Rawi and Hossam.
There's a question here that I think you would like to answer, whether if the virus undergoes some mutations, how accurate do you think your devices would be, and at what rate?
Yeah, I will take this answer because take the answer because our approach in our device, it's called cross-reactive sensors, in the sense that none of the sensors is selective by the so-called lock and key behavior. Our selectivity or sensitivity is reached throughout the training by algorithms or artificial intelligence. So whether we will have differences in the genetics or genetic mutation, we will be always able to again and again train our device for the new mutation and to take it into consideration.
Naama,
you want me to answer as well? So in our case, the detection is the nucleic acid. So the primers, it's small DNA parts that we designed for this method, aligned to the virus DNA. So if there are a few mutations, it wait, Rawi?
No, you go for it.
A few mutations, it doesn't matter. But if there are chunks of a lot of mutations or chunks of DNA that change, then we will need to design new primers. However, it's very cheap, the primers, and it's a process of a few days. I can say that also with this method, we can detect the epidemiology. So we can say you are infected and you're infected by the virus that its lineage came from this location or from that location. So I hope I answered the question.
Another interesting question to Professor Haick is, in asymptomatic patients, there is no inflammation response, and the metabolic changes of the VOCs, like what would produce these altered changes in the VOCs? You can see it's the last question here.
Well, the only people that can ask difficult questions are your previous PhD students. So this is Dr. Murad Nakhle from UK, and it's a very nice question. This is correct that it's rather challenging at the beginning to follow up the asymptomatic changes. We haven't done it yet. We are in the process to do so. And this is really very complicated experiment. And our assumption right now is that the human body response, according to the initial symptoms, will be too sensitive, so we can detect them in the breath at very low concentrations. We haven't done it yet. We are in the process clinically, and hopefully our assumption will come true.
Thank you both.
Thank you very much.
Are there any questions you saw and you'd like to answer and you haven't answered yet? You can take this time for that. I think now Professor Haick has answered the question about someone. He asked a question about the asymptomatic patients, and I think you just answered it.
Yes.
Yeah, I think we covered most of the questions. I hope I'm not missing anything. So at the end, I would like to thank you both for giving us these beautiful presentations. I'd like to thank you all who have joined us tonight and tuning in to listen about these amazing works. I also would like to thank the Technion UK to make this event possible and organizing it. And most importantly, I would like to thank you all for donating towards the emergency fund established to fight COVID-19. Thank you all very much, and I hope you had a good time. I hope it was interesting. Have a good night.
Thank you.
Thank you very much.
Bye.
Bye-bye.
Bye.